Imagine holding a fragment of ancient wood or a minuscule seed, eager to uncover its story and exact age. How do archaeologists transform these fragile botanical clues into precise chronological markers of the past? Radiocarbon dating works on plant remains by measuring the radioactive decay of carbon-14, or C14, within their organic tissues. This method reveals the moment the plant ceased its metabolic functions and stopped absorbing new carbon from the atmosphere, effectively providing a death date. The core principle lies in the unstable nature of carbon-14, a radioactive isotope produced naturally in the upper atmosphere by cosmic rays. Living plants continuously absorb carbon dioxide from the air through photosynthesis, incorporating both stable carbon-12 and the radioactive carbon-14 into their cells. However, once the plant dies, it stops this carbon exchange. The carbon-14 within its tissues then begins to decay at a constant, predictable rate, known as its half-life, which is approximately 5,730 years. By comparing the remaining ratio of carbon-14 to carbon-12 in the sample with the known atmospheric ratio, scientists can accurately calculate the time elapsed since the plant's death. To apply this in practice, archaeologists carefully collect plant samples from well-documented archaeological contexts. These samples undergo rigorous chemical pretreatment in a laboratory to remove any modern contaminants, such as soil humus or preservatives, that could skew the results. The purified carbon is then typically analyzed using an accelerator mass spectrometer, or a MS, which directly counts the carbon-14 atoms present. The raw radiometric age obtained is then calibrated using a global curve to convert it into a calendar age, accounting for fluctuations in atmospheric carbon-14 levels over time. What makes plant remains particularly valuable is their direct uptake of atmospheric carbon, which often provides highly accurate dates that reflected the immediate local environment. Short-lived plant materials, like seeds or small twigs, are especially prized as they offer very precise dates, minimizing the oldwood effect, where older inner rings of a long-lived tree could give a misleadingly ancient age for a younger archaeological event. Understanding these nuances helps reconstruct ancient agricultural practices, environmental changes, and even human diet with remarkable clarity. Ultimately, radiocarbon dating of plant remains provides an indispensable chronological framework for understanding past ecosystems and human interactions within them. It allows us to precisely date archaeological layers and associated artifacts, offering critical insights into the timelines of ancient societies, their subsistence strategies, and the environmental conditions they inhabited across millennia.